So previously we talked about in the last lecture about three different types of biological molecules, the three main biological molecules in our human body, which is carbohydrates, number one. Number two was proteins, and number three, we talked about lipids. Now we'll be moving on to food tests, that how are we supposed to test each of these biological molecule or the nutrient present in us, कि हमारे अंदर ये present हैं या नहीं है. For example, we'll be talking about a Biot test for protein, an emulsion test for fats and lipids, a starch test. फिर हमारे पास Benedict solution है, Benedict test है for glucose. So these four types of food tests will be included. First of all, we'll be talking about the test for glucose. Test for glucose में क्या है? Number one, add Benedict solution. A Benedict solution is basically a reagent. ये एक reagent है that you will be given in a lab. अभी आपको लैब में दिया होगा and you have to add Benedict solution into a sample solution in a test tube. This is the solution that you are supposed to test. आपको सर ने कहा है कि Amar, why don't you let me know whether the sample solution has glucose or not? First of all, you'll add Benedict solution into the sample solution in the test tube. Then once you have added it, heat at 60 to 70 degrees Celsius in water bath for 5 minutes. आपने क्या करना है? You would take that sample solution test tube and then you're going to heat it at approximately 60 degrees Celsius to 70 degrees Celsius in a water bath for 5 minutes. Next, you're going to take that test tube out of water bath and then observe the color. If the glucose is present, the color would be changed from blue to orange or brick red. So if I repeat the whole experiment, क्या था? You are given a sample solution. This is the sample solution that you are told to check. Now you don't know whether this contains glucose or not. आप क्या करेंगे? You would add this Benedict solution. अब जब आप Benedict solution डालेंगे, the color would obviously be blue. क्या color होगा? The color would obviously be blue. Now, if the glucose is present in the solution, आपके पास क्या result आएगा? That the solution would turn brick red. अगर solution orange या brick red हो गया, this means that yes, glucose is present in the sample solution. So first step, add Benedict solution, then heat it. जब आपने add कर दी Benedict solution को, you have to provide heat as well. और heat कितनी होनी चाहिए? About 60 to 70 degrees Celsius. After heating, you would be getting this result. What would be a negative result? Negative result, if it does not contain glucose, would tell you कि हाँ ठीक है, अगर इसके अंदर glucose नहीं है, the solution would remain blue. If the solution remains blue, it means it's a negative test and glucose is not present in the sample solution. But if glucose is present in the sample solution, the solution would change from blue to orange या फिर brick red. Now here you can see we have a tube 1 containing water and Benedict solution. Tube 2 के अंदर है glucose and Benedict solution. Hence this is the kind of color you would observe, which is a brick red color. जैसे ईंटों का कलर होता है. Solution 3 आपके पास है sucrose and Benedict. Now this was sucrose. Obviously sucrose के अंदर glucose नहीं होता है. इसके अंदर glucose नहीं था, तो it remained blue. This is an example of negative test. This is an example of positive test of Benedict solution. So here we have the whole experiment. ये आपके पास है test sample. ठीक है, the yellow color is the test sample that you don't know whether it contains glucose or not. You're going to add Benedict solution. आप क्या करेंगे? You are going to add a few drops of Benedict solution. अब इस solution का color क्या हो गया है? Blue. Now you're going to heat this test sample in a water bath. और इसका temperature आप रखेंगे 60 to 70 degrees Celsius round about. Now boil the mixture for 5 minutes and observe the color change if a reducing sugar is present. Do remember this, at the end of the class I'll be asking you questions that how is the Benedict test carried out. So first आपने क्या किया? You took the test sample, added a few drops of Benedict solution, heated for 5 minutes. और कितने temperature पे heat किया? From 60 to 70 degrees Celsius. Some of the safety precautions that you can take is number 1, handle the test tube with tongs, and number 2, wear safety goggles or heat proof gloves. और time आपने कितना रखना है? Time would be approximately 5 minutes. ये आपके पास time है, which is approximately 5 minutes. A positive result would be if the solution turns red, and a negative test would be if the solution remains blue. Positive test बताएगा that yes, glucose is present. Blue negative test would say that no, glucose is not present. So here we have blue solution means none. All it is containing is the Benedict reagent itself. ग्रीन और येलो कलर बताएगा ट्रेसेस ऑफ रिड्यूसिंग शुगर, मतलब कि लेस क्वांटिटीज में मौजूद है, दैट द ग्लूकोज वुड बी प्रेजेंट इन अ लोअर क्वांटिटी. Moderate color batayega if you get an orange PPT. Orange PPT would let you know that it contains moderate amount of glucose. Whereas if you get a brick red PPT, this is the pure positive result telling you that there's a large amount of reducing sugar. Reducing sugar हम basically glucose को ही कहते हैं. We are talking about glucose. When I use the term reducing sugar, I mean glucose.